Hi there. I'm Isabella Torres, and I will be attending Arizona State University this fall. There, I will be starting my path towards a bachelor's degree in biomedical engineering. Now, I would love to tell you about what sparked my passion for biomedicine. I found my spark in my middle school science class when I was introduced to CRISPR, which was my first intro into the world of genetics. Let me ask you a question. What if we could rewrite the code of life itself? What if genetics could just be like writing a book? Imagine, if you will, you're writing a book and you want to find a specific sentence to edit. You could flip through every page manually, but that could take forever. Now, imagine having a magical bookmark that could instantly jump to any sentence you want to change. That's essentially what CRISPR does. But instead of editing books, it edits the instruction manual of life. CRISPR stands for DNA, clustered, regularly interspaced, short, palindromic, repeats. But don't worry about memorizing that mouthful. What matters is that CRISPR is a revolutionary tool that allows scientists to edit genes with incredible precision. CRISPR-Cas9 represents both humanity's greatest medical breakthrough and one of its most controversial scientific tools. While it seems far-fetched, CRISPR didn't start in any kind of fancy laboratory. While studying bacteria, they discovered that bacteria have their own amazing immune systems. While studying this immune system, they found that when viruses attack bacteria, the bacteria does something incredibly clever. It takes a snapshot of the virus's genetic code and stores that code within its own DNA, like keeping a wanted poster of the virus. The bacteria also keeps a pair of molecular scissors called Cas proteins nearby. If the same virus ever attacks again, the bacteria can quickly recognize it using the stored snapshot and deploy its scissors to cut up the virus's DNA, destroying the threat. Two scientists by the names of Jennifer Doudin and Emanuela Charpentier in 2012 realized they could hijack this natural system and reprogram it to cut any DNA sequence they wanted. It was like discovering that bacteria had been carrying the ultimate gene editing toolkit all along. Let's break down how CRISPR works using a simple analogy. Think of DNA as a massive library with billions of books where each book is a gene containing instructions for making proteins that keep us alive and healthy. CRISPR has two main components, a guide and a pair of scissors. The guide is the exact address of the gene you want to edit. It's like having GPS that can navigate to any specific sentence and any book in the entire library. The scissors are called Cas proteins, usually Cas9, which is the most common one. Once the guide RNA finds the target location, it signals to Cas9, here's our destination. Cas9 then makes a precise cut in the DNA, like cutting out a sentence from a book. But CRISPR doesn't just cut, it can also repair and replace. After making the cut, scientists can insert new genetic material, delete problematic sections, or simply let the cell's natural repair mechanisms fix the cut. It's like being able to delete a typo, insert a new sentence, or replace an entire paragraph in the book of life. The whole process happens at the molecular level, but it's incredibly precise. CRISPR can target specific locations in our 3 billion letter genetic code. That's like finding a specific word in a library containing every book ever written. Even from the beginning, CRISPR wasn't just a laboratory curiosity. It's already changed the world in remarkable ways. In medicine, CRISPR is offering hope for diseases are using it to that were once considered incurable by editing patients' bone marrow cells to produce healthy blood cells. They're developing treatments for inherited blindness, certain cancers, and even HIV. Imagine being able to fix genetic diseases by literally correcting the typos in a person's DNA. In agriculture, CRISPR is editing crops that are making them more nutritious and resistant to diseases and climate change. Scientists have developed wheat that doesn't trigger celiac disease, tomatoes with longer shelf lives, and rice enriched with vitamins that could prevent malnutrition in developing countries. CRISPR is also helping us understand fundamental biology. By editing genes in laboratory animals, scientists can study how specific genes contribute to diseases, aging, and other biological processes. It's like being able to change one variable at a time in the complex equation of life. Environmental applications are emerging too. Researchers are exploring ways to use CRISPR to help coral reefs survive warmer oceans, create mosquitoes that can't carry malaria, and even bring back extinct species by editing the genes of closely related animals. The future of CRISPR is both exciting and requires careful consideration. Scientists are working on making CRISPR even more precise, developing new versions that can edit genes without cutting DNA at all, and creating systems that can edit. One of the most promising developments is using CRISPR directly in the human body, instead of removing cells, editing them in a lab, and putting them back. Scientists are developing ways to deliver CRISPR directly into organs that need treatment. However, with great power comes great responsibility. CRISPR raises important ethical questions, especially when it comes to editing human embryos or making changes that could be passed to future generations. There has also been some concerns over current accuracy rates and off-target effects, like mosaicism, which means, in simple terms, a mutation where something or someone has cells of different genetic makeup present in their body. But scientists are working every day to make sure these effects are less and less present. Society is still working through these complex issues, balancing incredible potential benefits with the need for careful oversight. The technology is also becoming more accessible and affordable, which is democratizing genetic research, but also requiring new safety protocols and regulations. Started as bacteria defending themselves against viruses, has become humanity's most precise method for editing the code of life. As we continue to refine and responsibly develop this technology, CRISPR promises to help us solve some of our most pressing challenges, from genetic diseases to food security to saving the environment. The age of programmable biology has begun, and CRISPR is writing the first chapter.